with Professor Andrew Palmer, who is Associate Professor of Biology at the Florida Institute of Technology. Andrew got his PhD in biological chemistry from Emory University in 2008, and he leads a lab of chemical ecology and astrobiology, which works at the intersection of biology and chemistry. His lab works on a range of topics, including chemical signaling between microbes, as well as developing new tools to regulate bacterial virulence. And today, Andrew, we talk about quorum sensing in Chlamydomonas. So without further ado, Andrew, thank you for joining us today, and the virtual floor is yours.
Great. Thank you all so much for the invitation. It's a real pleasure to get to talk to everybody. So as you can see, my lab is split between chemical ecology and astrobiology. And so it's really nice to come and give a talk about quorum sensing, which we talk a lot more about the astrobiology side here, where we live really next to, pretty close to Kennedy Space Center and NASA. So, when I was thinking about this audience, I thought it'd be really nice to actually talk about the origins of quorum sensing. But before I get into any of that, I'd like to thank, I always like to put my acknowledgements first, just so I don't lose track of all the people who participate. So quorum sensing is about working as a team. This is my current team here. So this is my current lab configuration. A lot of these people are working on the astrobiology side. And down below, you see all the names of the people who have been associated with the quorum sensing project, which has been in the lab for about 6 years now and is largely driven by undergrads. You have Kirsten Cutshaw, who is right here and is on this call. Kirsten was actually an undergrad here when she helped develop this project along with Tim Hare, who was my first PhD student, and Ali Folsick. And then together with Catherine Scholla and Melissa Riddle, who were here as part of a biomathematics REU that we had through the NSF, we helped really kind of develop this project into what it's become now, which is one of the sort of the second part of the lab, and it's really, I think, some spectacular work. And I can't thank them all enough for all the good work they've done. I'd also like to call out Chris Schuenard, who is a mass spec analytical chemist that we partner with at Clemson University. I'm not going to talk about any of that data today, but I'd be remiss if I didn't mention his contributions. And then Tatiana Karpova, who runs our electron microscopy facility downstairs, and Chara Bento, who is currently helping us with some of the metabolomic analysis, which I'm also not going to talk about today, but is part of the team. All right. So, and feel free to stop me at any time for questions as I go. So actually, the origins of quorum sensing actually start in the ocean. So with the discovery of the bioluminescent squid here, the bobtail squid. And so the bobtail squid is colonized by Vibrio fischeri. Can everybody see my my pointer?
Yes.
Yeah.
Okay, great. So sometimes that doesn't work. So Vibrio fischeri colonized this Hawaiian bobtail squid. And then what that results in is a phenomenon known as counter-illumination, where for predators looking up at the squid, it looks like moonlight on the water, so they don't go after the squid. And then there's ink sacs on top of the light organ where the Vibrio fischeri live. And that creates basically a dark spot that allows it to be camouflaged from predators that are floating above. So this is a really nice symbiotic mutualism that has evolved between these. And what's really fascinating about this was when they were first studying how this process worked, and I'll thank Bonnie Bassler, who put up a really nice online course with some of these nice images, is that they noticed an alternation between the lack of luminescence during the day when the bobtail squid was sleeping, and then the bioluminescence would come up over nighttime, right? And if you looked at the squids during that daytime period, even if you put them in the dark, they really weren't bioluminescent. And it was because they were populating up with the Vibrio fischeri over the daytime. So at dawn, the squid expels the Vibrio population and it goes and hides in the sand. Vibrios come back, and as you can see here on the right, they enter in to the squid and inhabit this light organ. And as their numbers increased, bioluminescence came up, right? But so there looked like there was some kind of connection between the number of microbes and actually getting bioluminescence to be activated. So that led the this experiment by Nielsen and Hastings that was trying to understand the relationship between microbial number and bioluminescence. And so there they took this away and did this in the lab. So we go in vitro and we start growing these cultures in a lab and looking at the accumulation of bioluminescence. And the real take-home message of this sort of cartoon figure here is that the rate at which bioluminescence increases doesn't match perfectly. It's not a linear relationship with the cell density. So you reached a cell density where all of a sudden bioluminescence was coming up faster, right? And so you're increasing the expression of the genes that were associated with that, right? And so that's really the first time that quorum sensing was actually documented. And that's over 50 years ago at this time now. So that paper is sort of like the classic in the literature, because after they'd said, well, okay, what is the source of this? And they hypothesized that there was something in the media itself that was allowing the bacteria to make the switch. So they took high cell density bacteria like this, and they centrifuged the bacteria out, they pellet it, they collect the media, and they applied the media to low cell density microbes. And sure enough, those low cell density microbes suddenly began to bioluminesce. And so it looked as if there was a chemical switch. Something in the media was controlling a phenotypic switching between the high cell density and low cell density states, right? Low cell density, no bioluminescence, high cell density, bioluminescence, right? So fast forward to where we are now, we've now determined that quorum sensing is something that happens in a large portion of the microbial world, especially the prokaryotic microbial world. Biofilms formation in gram-negative bacteria is heavily driven by quorum sensing phenotypes, and for instance, in Pseudomonas aeruginosa, but even biofouling on ship hulls, there's now growing recognition that quorum sensing might be playing a role in some of the initial colonization there. Nitrogen fixation in root nodulating bacteria is driven by this process. So legumes are able to form symbiotic associations, these little nodules that you see here that work as little houses in which bacteria known as rhizobia are able to fix nitrogen and provide that in a nutrient form for the plants. It's even an interkingdom gene transfer. So these tumors you see here are caused by the microbe Agrobacterium tumefaciens. And we've actually discovered that this is not just isolated to the prokaryotic world, but there are some examples, especially among fungi, in which quorum sensing occurs in eukaryotes. Other than Chlamydomonas, which I'm going to talk about today, there's not a lot of evidence that quorum sensing occurs in eukaryotes other than fungi. So Chlamydia is the only one so far. Mechanistically, we've also been able to go from the Nielsen and Hastings experiments and actually sort of determine that quorum sensing happens in one of two ways. In the first pathway, these signal molecules that were in the media, we call these autoinducers, that they accumulate and are either able to diffuse across the membrane or are actively transported, bind to some sort of transcriptional activator, like you see here, right? And then those bind to DNA and result in gene expression changes. Alternatively, these molecules may bind to some membrane-associated receptor. This version happens quite a bit in gram-positive microbes, and then that can lead to signal cascades and change in gene expression that way. Either way, the ultimate goal is that as autoinducer concentrations accumulate, they become like proxies for population density. And then once a certain concentration is reached, the system goes through a rapid change in phenotype. And so this becomes important when you're trying to juggle phenotypes that may not be beneficial when your cell densities are low. For instance, bioluminescence. If you only have 4 or 5 Vibrio fischeri cells around, bioluminescing is not going to provide a significant advantage to your host. If there's 100 million of you around that are bioluminescing, then that counter illumination effect can really be viable and useful, right? Same for making a biofilm. 1 or 2 microbes making some biofilm components isn't going to be particularly beneficial. When you have a bunch of them together, then that becomes a useful phenotype. So this allows us to control, or allows the microbial world to control phenotypic switching to densities that are useful. All right. When we look at the molecules that drive this, so I come back to my chemistry background, I'm always interested in the signals that are involved. We have, the most common molecules that we see are these gram-negative autoinducers here that belong to this one class of N-acyl L-homoserine lactones. And this R group has just a variety of different functionalities can be associated with it. We also have gram-positive autoinducers. And then, there's this group of eukaryotic quorum sensing molecules, but again, they are largely focused on fungi. And of Chlamydomonas and the fungi, we haven't found good examples of eukaryotic quorum sensing yet. However, this autoinducer here, AHLs again, they are very, very prominent. They're the most common quorum sensing signal that we see out there. And so that's where a lot of my research was focused, both as a postdoc and then when I started my lab here at Florida Tech. In fact, I kind of went in a slightly different direction. My original question, and this is what I was doing during my postdoc fellowship at the University of Wisconsin, was I was asking about how do eukaryotes respond to these quorum sensing molecules? So it seemed kind of unfathomable to me that all of these dialogues between potential symbionts, both mutualistic and deleterious, could be going on in the roots, in the rhizosphere, the area around the roots of plants, and plants not be able to detect it. And so we had a project where we were looking at how plants absorbed these acyl homoserine lactones from gram-negative bacteria, how they processed them, how they led to physiological changes. And we were leveraging a bunch of organic chemistry knowledge to make various, pro-drug-like analogs. So we were doing some classical sort of medicinal chemistry, structure-activity relationships, kind of probing activity in these systems, trying to understand what plants could get from this. And so then I moved for my postdoc and I started here at Florida Tech. And I had my first graduate student immediately said, I don't want to be a plant guy, which is kind of hard to say to the guy who was just hired as the plant guy. So I said, well, I don't really know what else. What do you want to work on? He said, well, could we work on something that still does photosynthesis, so it'll still be green? And I'm like, okay, that's kind of a reach, but let's see, what have you got? And so he suggested Chlamydomonas reinhardtii. And so Chlamydomonas is a great model system. It's genetically tractable. It's widely distributed ecologically. It's easy to grow. And it has its own microbiome. So we considered the possibility that there was some evidence also out there that they might have the ability to modulate quorum sensing. And I said, okay, well, maybe this is sort of a unicellular system where we could ask some of those similar questions that we're asking plants, and we could translate this. And so here you see sort of this artist rendition of Chlamydomonas. And then here, these are electron microscope images that actually we've collected. You can see they have these two flagella here. And they're capable of photosynthesis. They're very simple organisms, but what's really nice about them is a lot of the physiology of this organism is very well defined. Their photosynthetic mechanisms are well defined, motility is well defined. So it gave us a lot of tools that we could probe molecularly what was going on and have some confidence in what our results might be and what we could compare them to, right? So where we started was with a very simple question. We tried growing them in different AHLs and those acyl homoserine lactones from gram-negative bacteria. And that's pretty much where our AHL story here is going to stop, because this is where the story sort of diverges. There's a part of the lab here now that's still looking at how Chlamydomonas responds to those signals. And here we are 5 years later from when we started that project. We're actually putting together that paper right now. But I want to really talk to you today about the discovery of Chlamydomonas as having its own quorum sensing system, right? And that's what's really been exciting. So one of the thoughts we had was, well, maybe AHLs could influence growth, and we were testing that. And I said, well, what's another readout we could do that'd be really simple? And at the time, I was still trying to get my lab going. So we were finding projects for undergraduates to work on and new technologies. And I said, well, maybe these acyl homoserine lactones can change the swimming speeds of Chlamydomonas. And I said, well, how do people figure that out? So we looked in the literature and we found that the way that most labs were determining the swimming speeds of Chlamydomonas was you take your culture, you put it on a slide, and then you count. You basically find a cell and you track its movement from point A to point B with a stopwatch. And then you go do another one, a third one, and you collect 10, 12, maybe 20 of these for your sample. And then you average that together and say, they move at about this rate. So we went, okay, well, at that time we had this biomathematics summer research program, an REU through the NSF. And we said, well, can we do better than that? And so we started talking to them, the microbiologists, so myself and a couple of the mathematicians. We said, well, how well do a dozen counts actually approximate the swimming speed of the cultures, right? So what happens when a cell leaves focus? So it doesn't travel that distance. So do you stop counting that? So, and motility is often stochastic. So, by only looking at 10 or 20, are you really capturing a good distribution of the population? And then we really started thinking about the concepts of statistical power and confidence. And if you have a culture of 10 to the 7th cells per milliliter, and you're trying to get a really good measurement of how fast they're moving, counting 10 or 20 is a really small sample size, right? And there's a lot of bias. We found that when we started collecting the images just by hand, that there was a lot of bias that creeped up into which ones we chose to follow, which reinhardtii we watched swim. So this is how we got to using TrackMate. So TrackMate is a plugin for ImageJ that's meant for following movement. We've learned over the years that we are pushing TrackMate up to about its computational limits, because we are taking cultures that are 10 to the 5th, 10 to the 6th, even 10 to the 7th cells per milliliter, and we're trying to get TrackMate to follow them. So the process is inherently starts the same. We take a culture and we put it on a microscope slide and we collect a video. And then we run that video through some processing, some pre-processing, and then through the TrackMate plugin, which is part of ImageJ. And what we get out is something like this. And so here what you're seeing is each of these purple dots represents a single cell of Chlamydomonas. We're going to call Chlamydomonas clammy. And we're following its movement. And these lines are giving you the tracks, right? And so it's a lot easier to follow these, because if you're hoping to watch clammy move in a straight line from one direction to the other, that often doesn't happen. In fact, it can be a little wavy, right? So we get better distance measurements this way, but also we can acquire thousands of tracks in a 30-second video, right? So we collect a 30-second video, and then we allow this software to do the movement for us. We found that it can even distinguish between collections. So sometimes clammy cells will group together in tetrads, dyads, and triads, and we can actually distinguish those. So here's blowing up on one of those. We see a tetrad here. We see sort of a triad forming there, and we see sort of dimerized clammy cells there, right? Duos. So we're able to distinguish those groupings, and we're able to track these things over that time period and collect a lot of data. And so from that, we can build histograms, essentially, of the distribution of movement. So one of our first questions was, does this method actually bias movement across the slide, right? So in the absence of any external stimuli, movement across the slide should be random by Chlamydomonas. So what we were able to do is run things into ImageJ. Sorry, I'm moving picture out of the way here. And so, wait, I skipped a slide. There we go. Sorry, I jumped ahead one. We take our slide and we're going to look at this from the back of the slide to the front of the slide. You could call it top and bottom, whatever you will. We run it through ImageJ, and then we export from ImageJ and put into R essentially all the vectors that the algae were moving in, right? So we're saying just where does Chlamydomonas swim? We break it up into 8 sections on the slide that they can be at. And what we see is there's no bias here in where they're moving. So they occupy all the sections, and all the tracks are equivalent throughout that entire slide. So it's telling us that we're not biasing the directionality just by having them there. And we said, well, that's great. Is there a way that we could show that by actually intentionally biasing it? So it's well known that Chlamydomonas displays phototaxis. And so what we did here was we do the exact same experiment, except this time we shined essentially a pen light across this way. And so we create a light vector like this, where the extra light is coming in this way from the back of the slide, right here where you are, where the user is, right, and up the front slide. What we see is the tracks bias themselves to that region. So now the clammy line up along that path of light, and that's where they're spending their time when they're swimming. So this means that we have a really good control over this. It also means that in the absence of any external light, the Chlamydomonas do seem to be swimming at least in a normal fashion. We're not biasing their swimming too much by just having them on the slide. So this is kind of a nice experiment that showed that we could control this, and that actually those subtle differences could come out in our data analysis. All right. So what do we see when we look at this? So what we do is we run an analysis like this. We take the raw data, we plug it in, and we generate a histogram where we bin the different track rates like this, right? So each of these represents the number of the total counts, and each of these usually have several thousand counts associated with them of different track velocities, right? And so from this, we're trying to get a distribution of how the population moves. And so what we found in the literature is the literature was usually reporting that these things moved somewhere in this range. And what we saw was slightly different, right? So part of that could be the methodology that we use, but it's also saying that there's a bigger population distribution here, right? And that perhaps looking at these movements in terms of an average is not always the best way to think of this. We want to see the population as a whole. So this was really important. We'd spent a lot of time building this methodology up. It took us about 2 summers working with the biomath REU and then some side work along the way to get here. And then I said, okay, well, let's put this data together with the AHLs and let's get going. And one of the students in the program said, well, we're waiting for the cultures to get older. And I said, why? And they said, well, they move faster when they're older. And I said, what do you mean? And so they showed me this data. And so here we're looking at cells. This is a still of the 24-hour, 48-hour, and 72-hour cultures. And then this is basically a snapshot of that map of movement, right? So we're watching more and more dense cell movement across here. And then down here, we've got the bins. And so here's at 24 hours, actually about 40% of the cells aren't really moving much at all, right? But at 48 hours, which is the graph I showed you previously, we start to see some acceleration. But look what happens at 72 hours. We really start to move towards this sort of Gaussian distribution of cell movement here, right? And so they definitely sped up. I said they sped up as the cultures got older. And we thought about that for a day before we said, no. They didn't speed up because the cultures got older. What if they sped up because the cultures got denser? The phenotype changed because the cell density increased, right? And so here is actually the cell density measurements that go with those time points. So at 24 hours, we're looking at about 2 times 10 to the 5th cells per milliliter, 5 times 10 to the 6th cells per milliliter at 48, and 2 times 10 to the 7th after 72 hours, right? So that really was where the project changed. And we diverged completely from wondering about what AHLs were doing, because then we said, what are these things doing? How is this possible? So we went back into the literature and said, well, what did people do originally? Well, that media swap experiment worked for those guys, so maybe it'll work for us. So we did the same idea. We took high cell density, our 72-hour cultures, that 10 to the 7th cultures. We centrifuge and pellet it. We remove that supernatant. We filter sterilize the supernatant, so there's no cells there, right? We take that conditioned media, add them to low cell density, right? And say, well, what's going to happen? And our hypothesis is, this is quorum sensing. They should speed up. So this is that experiment. So here what you see are white bars, are 24-hour cultures, just left alone. In the gray, we have 24-hour cultures that we spun down, resuspended in the same volume of 72-hour media. So the media that was preconditioned, sterilized, and then added to that 24-hour media. And sure enough, in the gray, you can see after about 6 hours, we get a really nice reconstruction of that effect, right? We basically induce these low populations to swim faster, right? So we've induced that high cell density effect. So with that in mind, we said, well, okay, there could still be something that's accumulating in the 72-hour culture. We can't rule that out. So if it really is something like quorum sensing, you should be able to artificially induce that. And in fact, there's been some really nice papers in partnership with cell biologists and chemical engineers who suspend quorum sensing cells in microdroplets. And they show that even a single cell of something like Vibrio fischeri will quorum sense if the cell density, if the space is small enough, so that the cell density is high enough, right? So we basically try and mimic that here. So we take low cell density cultures, we pellet it, and then we resuspend it and in a much smaller volume. So we do a 100-fold increase in the culture. So if we have a 10 to the 5th culture, we're essentially increasing it to a 10 to the 7th culture. Then we resuspend it in TAP, which is trisacetate phosphate. That's the buffer that we use for growing Chlamydomonas. Resuspend it, and then we come back in 2 hours and say, well, how's that affected the swimming speed? So here in the white bars, you see the untreated. Again, they're not swimming very fast. In this concentrated experiment, though, right, where we concentrate the population, you can see that we've mimicked that high cell density effect again, right? So we've created a high cell density environment in much less than 72 hours, and the process kicks off and it begins to swim faster. Our first thought was, well, what if that's just the act of resuspending the cells? So we did a resuspension control right here and this bar, and you can see those don't have a significant effect, right? So it's not just that we vortexed the cells and resuspended them in solution. There's definitely an effect of concentrating the cells that recreates this effect, which is very consistent with quorum sensing. All right. So if that's the case, we should be able to isolate a quorum sensing signal, right? So we can take cultures of Chlamydomonas, and so we'll do an old separatory funnel phase extraction that way. And then we also do a solid phase extraction method. We started with the organic extraction just because that's, older chemist, that's what I was trained with. We've since moved almost exclusively to using the solid phase extraction method, mostly because it gives us much more consistency across different runs. It's also more compatible with cleanup for methods that we want to use downstream, like mass spec and some of these other things. And it's also, long run, actually becomes cheaper than using all these organic solvents, and better for the environment also. And the users, since you don't have to, go through a liter of hexane, ethyl acetate, and whatever else. All right, so from both of these, either solid phase extraction or from here, we're going to evaporate that off. We redissolve in DMSO in a very small volume. And so we've gone from 250, 500 mils of cultured, of conditioned culture, what we call high cell density material. And then we end up with extract that we can go ahead and test. So we go ahead and test that. So here in the white are the low cell density, right? So at 24 hours, you can see this is their growth or their swimming speed. If we take high cell density, this is swimming speed and high cell density. And then here in the gray is the low cell density, so 24-hour cells that we've exposed to our high cell density extract. And sure enough, those show a speed up, as you can see from these green arrows. Right? So since then, we've gone through and we've tried to do some characterization of the molecule. We know that the molecule is pretty stable, so we can keep it in the freezer for over 6 months. It's shelf stable for at least 1 month. That was done by accident when someone forgot to put one of the freezer bottles away. We do know that it is a little heat sensitive, though. So if we keep it for overnight at 37 degrees Celsius, we get about 50% loss in activity. We're actually tracking on pH sensitivity right now. So that's something we're working on. So, obviously the question that we really want to get to is what's the molecule, right? So our first attempt, this was work that was done by an undergrad, Joseph Good, who is now at University of Central Florida working on his PhD on a different project. Joseph went through and along with Kirsten and a couple of high school students we actually had visiting the lab, went through and tested a number of known quorum sensing molecules and said, well, could it be one of these? So we've tried farnesol, tyrosol, tryptophan, and phenylethanol. These are all molecules that are involved in fungal quorum sensing, and none of them have any effect. We've also tried acylhomylserine lactones, and that doesn't seem to have any effect. We're going to be trying a couple of these others soon, but there's no literature that suggests that any of these other molecules are actually made by Chlamydomonas either. So it really seems like the Chlamydomonas quorum sensing is likely novel. So where the project is going now, and this is what Kirsten's spending her last year working on, is trying to work on the identification of this molecule. And that's not really an easy feat. So at first we thought, well, how complicated is the extract of Chlamydomonas reinhardtii, the what we call the exudates in the plant world, the molecules, the carbon chains, the different material organic compounds being released by Chlamydomonas out into the growth media. So it turns out there's well over 100 compounds in that mixture. And we don't really have a good sense necessarily of the bioactivity of the molecule, or even if there's only one molecule, there could be multiple molecules. So we're calling this the search for the clammy swimming speed factor, or the CSSF. And this schematic here is basically showing you sort of the two-pronged route that we're taking to try and get this molecule. So on one end, we're doing some routine HPLC methods, so going through rounds of fractionation, testing those for activity, harnessing the active fraction and going back. And so that's kind of allowing us to hone in on a subset of molecules that look like they have activity, so that we could go and do more structural identification on those through like mass spec and NMR. We also have, and this is our partnership with the Chouinard lab at Clemson, where we're actually doing a mass LC-MS-MS approach. So we're basically doing a large metabolomics assay of those exudates, the things that are released by clammy into the media, and basically looking for target molecules that have different concentrations between low and high, because we know that quorum sensing molecules should accumulate to a greater degree in higher cell density cells. And so those are two prongs, and I'd be happy someday to come back and tell you more about that, but I just kind of want to let you know where we are in the search for that. So another thing that we've learned about this I think is really interesting is that whatever this switch is, it's really dependent upon the production of new proteins. So this is cyclohexamide, which you can use to inhibit ribosome activity. If we have no extract, again, you see we have our regular activity like this at 24 hours. If we add extract without cyclohexamide, we get our normal turn on and our cells speed up. If we pre-incubate them with a little cyclohexamide before we add the extract, there's no effect, right? And we've tested this concentration of cyclohexamide. We know it's not lethal to the cells, but it's pretty clear that the switch probably requires the production of new proteins. And so right now we're starting to look at partnering with a lab here on campus that actually is a new proteomics faculty member that got hired, so we could do some proteomics work on this. How broadly distributed is the phenomenon? Well, we went out of reinhardtii and we checked one of its closest cousins. So this is Chlamydomonas moewesi. And so if we look at moewesi activity, this is not Chlamydomonas reinhardtii. This is Chlamydomonas moewesi. You can see at 24 hours, it doesn't swim very fast either. At 72 hours, though, it does speed up. And if we do the same kind of extraction procedure that we did for reinhardtii, we can recreate that effect really, really nicely. What was even more intriguing to us is that if you swap extracts, they still work. So the extract in C. moewesi, if you expose C. moewesi extracts to C. reinhardtii extracts, these are statistically indistinguishable, right? And so that's telling us that the extract, the signal molecule, that clammy swimming speed factor, is actually conserved, or at least structurally similar between these two species, which is really intriguing. So we're actually trying to build on that work now to leverage natural variation in quorum sensing to sort of understand the mechanism, right? And so the thinking on this, right, is that if we look at prokaryotic quorum sensing systems like Pseudomonas aeruginosa, the Vibrio species, even E. coli, not all strains within the species have the same quorum sensing responses. And so some of them have quorum sensing circuits, others don't. And so the question is, could that kind of natural variation be used to provide us about insight into quorum sensing in Chlamydomonas? Well, going through the literature, it turns out clammy wild type variants are actually very, very different. And so there's a large amount of genetic diversity in the wild type isolates. They have different rates of iron uptake. You have different cell sizes, different growth rates, different swimming speeds. So all of those are factors that we could essentially use, right, those features to see if we can use this process to identify other phenotypes that might be under quorum sensing regulation, or even a better understanding of the molecular mechanisms, right? So what genes are involved in this process. So, go ahead. I don't know if that's okay. So just sort of in a graphical display, we can take all these different wild type variants. We can grow them up to high cell density. We can look at their different extracts, and we can do our typical speed assay with them. But then we can also do extract assays where we do extraction. We've kind of moved away from the media swap because that experiment is actually it's really time consuming. It's actually faster and easier for us to do solid phase extractions. And then we have all the extracts. And then from the extracts, we could actually try swapping them between different wild type variants to see if they have activity. So this project is still pretty new. So I'm only going to talk about a couple of results from it, also in the interest of time. So here we're looking at CC1010 and CC1690, which are both, again, wild type variants of Chlamydomonas reinhardtii, as opposed to the 124, which is the one we've done most of our work on. That's one of the kind of major lab strains of that organism. And so these have different growth rates. Both of these grow slower than the 124. So we're having to move that time window out. But if you look at CC10, 1010, you can see at 48 hours, we're not really seeing any speed up effect. But sure enough, at 96 hours, quorum sensing seems to have started, and we're getting an acceleration of swimming speed. We actually just saw this data yesterday for 1010 and 1690. In 1690 at 96 hours, we're also starting to see something that looks like a speed up. They're actually, students are going to be in the lab this coming week, basically redoing this experiment, see if we get the same thing, and also working on extract isolation so we can go ahead and try and artificially induce swimming speed acceleration. So that's two strains that are kind of behaving like what we expect. But then there's this other strain that we work with called 4414. And 4414 is one of these Chlamydomonas strains that was isolated in a snowbank. And so they grow much, much slower, and they don't really seem to swim very much at all. And so this is a project of one of the master students on this project, Eli. And Eli was looking at whether or not this organism, this particular variant of Chlamydomonas, was even able to quorum sense. And so he started looking at low cell density and with high cell density extracts. And so one of the things we noted really quickly is that the low cell density hardly swims at all. If you add high cell density extract to it, that doesn't change its swimming either. I don't have the data on here, but I can tell you that even at 96 hours, it doesn't show any sign that it's sped up. It just continues to swim very, very slowly, if it even moves at all. A lot of the times it just sort of sits on the slide and maybe kind of shuffles around a little bit. We even tried taking 4414 low cell density and adding our 124 extract to it to see if maybe this organism doesn't make the molecule, right? So maybe it doesn't make, this particular strain doesn't make the quorum sensing molecule, and that's why it doesn't swim fast. And nope, we can't get it to turn on either. Now, what's cool is we can take extracts from 4414 and put that on 124, and that doesn't have any effect either. So we think with this organism, what we've discovered with this particular strain is we may have discovered the first of a strain of the wild type that doesn't make the quorum sensing signal and may not be sensitive to it either. Right. And so if that's true, we could actually go in and do some genomic work here to try and figure out what are the genes that are associated with the process of quorum sensing production and quorum sensing molecule detection in the system. And so this is really very interesting to us, and we're kind of following the leads on this right now. So that's our quorum sensing story right now, right? I think the discovery of quorum sensing in Vibrio fischeri, right, that discovery over 15 years ago resulted in a massive change in our understanding of microbial behavior, microbial ecology, and it had huge impacts to multiple disciplines. Now, quorum sensing is something that's being thought of in terms of therapeutics. We are thinking about it in terms of how to, we have a Center for Biofouling Corrosion here, and I'm working with one of the faculty in there, trying to look at whether or not you could inhibit quorum sensing as a way of limiting biofouling from initiating on ship hulls. And, we think about it in terms of wastewater management. We think about it in terms of plant growth promoting microbes, forming biofilms on the roots of plants, right? So it's had huge impacts across multiple disciplines. And now quorum sensing appears in Chlamydomonas, right? Which is this widely distributed genera with very large biological or biotechnology and ecological significance, right? And so Chlamydomonas is used as a model system for thinking about the production of biofuels, lipid production, understanding photosynthesis. And this is actually the first non-fungi quorum sensing system that we've found in eukaryotes. And so, my final thought here is I think that what this is showing us is that possibly quorum sensing is far more prevalent in the microscopic world than we originally thought. And I think, we not just solely because of this work, but I think we're poised to make another big discovery in the eukaryotic microbial world that says this sort of hybrid multicellularity that comes from quorum sensing, coordinating behaviors across these individual microbes could actually be more prevalent and more important than we think of. So it could be sort of a hidden component in a lot of the microbial systems that we're trying to understand. So with that, I'll thank you for your time. I'm very happy to answer any questions you have.
Thank you for your talk, Andy. Questions, Sophie?
Very nice talk, thank you. That's super interesting. I just had a question, maybe more of a hypothetical one, but you said at the beginning, like these, like bacteria, they would only quorum sense to build a biofilm when there's others around, or bioluminesce when there's other around. Do you have any ideas of why clammy might, like ecologically, why it would make more sense for it to swim faster if it has others around it?
Yeah, so one of the ideas here is, and I didn't talk about it, maybe I should use that example. Some of the microbes that also quorum sense do it for swarming. And so it's like, for instance, in Staph aureus, sometimes quorum sensing is a sign that there's too many microbes around and you might have to start competing for nutrients. So you disperse, right? And so one idea we have is that this might be about a recognition that cell density is getting high, so there's a risk of nutrient scarcity settling in. So perhaps it's time to move to new territories, which obviously in the microscope slide, there's not very far for them to go. We've tried doing a little bit of nutrient taxis in that idea, like providing nitrogen sources, but it's already known that Chlamydomonas does that. So it's a known phenotype that they have. So we're trying to figure out how we could do that test, like kind of in the lab.
Thank you.
Marco.
Yep. Hey, Andrew. We don't know each other. My name is Marco Polina. I work in a research institute in Spain. So thanks for the talk. I'm a physicist, full disclosure. So I have sort of like almost, I don't know if it's a philosophical question or not, but I just wanted to have a little bit your idea as to what exactly is meant by quorum sensing. And the following, let me see if I can explain myself. So the quorum sensing that I know so far is the one that you described that generates a change in gene expression. Like, for example, for the swarming, there is a switch to the swarming phenotype, right? Okay. So that doesn't seem to be the case here, as far as I understand. on the other hand, what we could have, so sort of the analogy that I was thinking about is the following: if we are all in a closed room, if we have a bunch of people in a closed room and we keep on breathing, at a certain point the CO2 goes up and then we die. Okay, we suffocate, so our motility goes down. And in a sense, I wouldn't call that quorum sensing, right? I would call that a result of our modification of the environment. So what would be the evidence that in your case, or how can we distinguish in your case, the first case from the second case?
I think it's the fact that you can take isolated signal molecules, so you can make an extract, right? And so it's not just nutrient depletion, because I can take an extract of the 72-hour culture and apply that to a 24-hour culture.
Yeah, but it doesn't need to be nutrient, right? It could be, for example, some other waste molecule that is still there that I am sensitive to because of some reason. And I don't know, I don't understand whether that would count as quorum sensing or not. In a sense, I don't really have a. This is sort of at the boundary of what I can decide whether it is with confidence whether it is or not quorum sensing.
No, so I would agree with you. This is something we discuss in the lab a lot. Is we call it quorum sensing because it's a density dependent effect, right? So one of the things that we've really been trying to work towards, and part of this would be identifying what the molecule is and whether or not it's something that accumulates exactly like how you're speaking, right? Is it a waste molecule or is it a specific signal molecule, right? So one of the things about quorum sensing that we don't have 100% on the answer to, right, is so far we've ticked a bunch of the boxes, but what would be nice is if we can identify the specific molecule and connect that molecule specifically to this effect, right? So AHLs, for instance, in gram-negative bacteria, they're made for that purpose, right? And so this is there's a universal quorum sensing molecule, AI2, and there's questions about whether AI2 is really a quorum sensing molecule because it's used for other things also. And so I think that's a really good question, right? So we're calling it quorum sensing now because that's the best label that fits it. But yeah, I think, you know, this getting the molecule is really the kind of the last part of this puzzle to really say that. So I would say, you know, can I definitively say it's quorum sensing? No, I would agree with you that there's room for a question mark there.
Sang-Yoon?
Hi, thank you for the talk first. Yeah, I'm a mathematician, so like I'm looking for the model that's so like it looks like I think it's also a follow-up question for like Marco. So like I am still confused that like you said like the speed of the molecules, they're like moving faster when their density is higher. Is that in a free space, or like is it like it's captured in some um it has a limited capacity, or is it like just a free capacity so like they can actually move like freely?
Well, so the microbes, right? So the chlamydia are within the slide, so they're on the slide itself, and they can move out of our field of focus and then come back into it. That happens. But so, you know, that within the size of the droplet of water, I would say that that's certainly not an infinite space, but it is a large space even for a unicellular eukaryote. It's still quite a wide space for them to travel in. Yeah. So.
And then sorry, yeah. And then the course they try to track for like their like, so they try to track for their food, so like they wanted to feed them. That's why they're trying to go like to expand like faster.
Yeah, so the our hypothesis is that the movement is related to basically the idea again of dispersal. So you get to a signal that there's a high concentration around, and that leads to dispersal. Now that gets tricky, right? Because that gets into, are you just responding to a waste material, right? So that's where we're on the fine line there. But I think it tracks with what we see in some other organisms that quorum sense, where when you get to a high cell density, it's time to disperse and move on. It's just in this case, they're trapped in the slide, so they only can just swim faster, right? We're not giving them an infinite direction to move in. It'd be really interesting if we could get a system together like that. One of the things we've been trying to figure out is a sort of a soft auger system where we could get better movement, but clammy doesn't grow very well in those kind of contexts. Like a swarm auger. Yeah.
Okay, thank you.
Yeah.
I have a question regarding the critical concentration, which triggers the speed up. Maybe I missed that. What is that concentration, and how does it compare to some scenarios in real environment?
Well, so that's kind of our question, right? So in the absence of the molecule, like a real molecular identity, we're adding extracts, right? So we don't know from run to run how much exactly is in the extract. That's why there's such a pressure on us right now to get the molecule, right? And I think that's really the big next step for us, is to get the molecule, because we can answer those questions about dynamics. We know that we have a set concentration. We're usually adding about 5 microliters per milliliter of our extract, to the culture in order to get it to do the speed up. But we don't know exactly what that corresponds to in terms of molarity, because we don't have.
But the density of the clammies, right? You grow them and they divide, and at some point they will reach a concentration high enough that presumably they will produce high enough concentration of the unknown compound that will trigger the speed up.
Yes.
So what is the typical concentration of the cells at which that transition happens?
So we start to see it really around 10 to the 6th. In the 124, which is the lab strain. It looks like I'd say right now with the variants that we're seeing, it's usually around 10 to the 6th that we start to see it. The problem is some of the cultures grow slower, so relying on time measurements again turns out to not be useful. We have to really rely on the growth measurements. So we have to track growth and then correlate growth to speed. But I'd say 10 to the 6th is the range where we start to see the speed up.
So perhaps ecologically speaking, this could be a bloom condition, like algal bloom condition.
Under what condition?
Bloom condition.
Yes. Yeah. Okay. So, yeah, I don't. So a lot of times, like clammy living in the soil doesn't have that same kind of situation. Like blooms don't really happen the same there. But so it's possible though that you can reach these concentrations. But yeah, so it probably doesn't happen an awful lot, but that's actually one of the mistakes that's I think was made in the quorum sensing community. Was we ran out and found a bunch of examples of quorum sensing, but we need to realize that quorum sensing probably represents what happens with Vibrio fischeri is pretty unusual comparatively. Things like a lot of the other quorum sensing systems happen for like 1% of the microbes 1% of their life. And even in Vibrio fischeri, right? If you think about it, when the squid expels a lot of the Vibrio fischeri back out into the water, those are generations of Vibrio fischeri that never quorum sense, right? So it's held onto by a very small amount of the population. So when this effect occurs, it's important, right? So then we see this for nodulation, right? There are rhizobia bacteria all in the soil. Only a fraction of them will ever form a nodule and be associated with a root. But when it happens, it's critical. So, that's it. It's really about the fact that not everybody's going to do it, but you have to have the mechanism in there to do it. Right? So it's beneficial.
Thank you. Thank you. There's a question in the chat. What is the maximal cell density where you can track cells and measure the speed?
Ooh, I don't I don't know the answer to that. I don't think we're right now experimenting with some, we're playing a game with the ones that grow really slow. The 1010 and the 1690, they both grow really slow. So we're trying to do some games where we're basically adding extra nitrogen to see if we can kind of redline them to make them grow faster. And we wanted to see if that was something we could use. So that's an interesting question. What's the upper limit? We don't know. Yeah. I'm gonna make a note of that. I want to know the answer to that now. When do we crash the computer?
I mean, your student Kristen provided the answer that you got up to above 10 to the 7, maybe. Okay. Are there any other questions?
If we can go much further than that, I'd be really surprised.
Are there any other questions? No? Oh, there is one more. Okay, we will take one last question from Luis.
Yeah, sorry, I was just wondering about if you see any similarities with the aggregation behavior we seen in Dictyostelium discoideum, for example, the role of CAMP in this transition to higher swimming speed?
Do I see any correlation? We haven't looked at that. That's a really that's a good idea. We thought about it at least once, and I think I don't have a student free to work on that yet.
Okay, thank you.
All right, I think it's almost 6 o'clock. It's time to stop. Thank you, Andrew, for the very engaging talk.
Thank you.
And before we finish, let me just remind you that we meet again on October 24th, when our speaker will be Daniel Repeta from Woods Hole. Thank you very much to everybody.
All right. Bye. Thank you, guys.